Affimers are an alternative to antibodies, which are the gold standard affinity reagent used right across life sciences. $50 billion worth of market, despite some serious limitations. So there's a huge unmet need for alternatives to antibodies. And Affimers are Avacta's proprietary engineered alternative to antibodies that have none of those limitations.
So antibodies have been a boon to life sciences researchers, and there is no way that life sciences research would have progressed to where it is today without antibodies being available, but they do have the disadvantages. I think it's fair to say that an unacceptable proportion of commercially available antibodies don't behave as expected, so they either cross-react with other target proteins or they just don't work in the assays that researchers want to use them in. That's a big problem if you're a researcher who's done a body of work and then finds that out after the fact. It does tend to invalidate some of your work, which is an issue. So we raise Affimers in a way which is quite different to the way you typically make an antibody. Affimers are generated using screening technologies which don't rely on an animal. It means we can introduce things during our screening process that drive specificity or might drive a particular KD, and it lets us generate a binder which has much superior engineered by choice performance, as opposed to an antibody where you do raise good antibodies by putting an immunogen into an animal, but it really is a case of see what you get after the fact. With our process we can design in what we want as part of the screening methodology. A good quality monoclonal reagent can take somewhere between 9 and 12 months to generate. We can go from a target protein or small molecule to having candidate Affimers within 7 weeks.
Our near-term focus is on generating Affimers as research reagents. This is a billion-dollar market in academia and in industry, but the important thing is there's no regulatory hurdles to overcome, so the time to get a product to market is very short. So we're addressing the research reagents market in 3 ways: through custom Affimers, which is a bespoke Affimer service where we generate Affimers to a customer's particular target, through an online catalogue of Affimer reagents, and also through strategic licensing of Affimers to partners who will put the Affimers into their products. Affimers have also got a huge opportunity in therapeutics. So what we're focused on now is generating a data pack that will allow us to license Affimers to third parties who have the resources to be able to develop and take Affimers through to market as therapeutics. Well, I think the thing that really excites people is that we're developing a really high quality set of tools that will give life scientists a chance to work smarter, faster, and hopefully apply those to real advances in medicine, in diagnostics and in treatment.